Ethanol metabolism modifies hepatic protein acylation in mice. Mitochondrial protein acylation increases in response to chronic ethanol ingestion in mice and is thought to reduce mitochondrial function and contribute to the pathogenesis of alcoholic liver disease. Ethanol is metabolized by alcohol dehydrogenases to form acetaldehyde and then by aldehyde dehydrogenases to form acetate, with subsequent formation of acetyl coenzyme A. Acetaldehyde is highly reactive, toxic, and high levels from excessive ethanol consumption can damage cellular proteins and DNA. In the mitochondria, SIRT3 is the major regulator of protein acetylation levels. SIRT3KO mice showed mitochondrial protein hyperacetylation, whereas mice lacking SIRT4 or SIRT5 showed no increases in protein acetylation. Under basal conditions, SIRT3KO mice are metabolically normal, but under stress conditions showed reduced fatty acid oxidation and developed metabolic abnormalities. Because acetylation has an important role in ethanol metabolism, it was hypothesized that protein succinylation could be an important post-translational modification regulating mitochondrial metabolism.
Materials and methods. The animals that were used were mice that were fed a modified liquid diet for 6 weeks before harvesting their livers for analysis. The diet contained 44% fat-derived calories, 16% protein-derived calories, and 40% carbohydrate calories or ethanol derivatives. Ethanol-fed mice began the study on a diet consisting of 2% ethanol, with the ethanol-derived calories increased on a weekly basis until sacrifice. After 6 weeks, the mice were anaesthetized via intraperitoneal injection of sodium pentobarbital and euthanized by exsanguination. The livers were then harvested and subjected to differential centrifugation using a sucrose gradient to obtain the fractions needed to perform the experiment. In Western blotting, the protein mixture is applied to a gel electrophoresis in a carrier matrix to sort the proteins by size, charge, or other differences in individual protein bands. The separated protein bands are then transferred to a carrier membrane. The proteins on this immunoblot are then accessible for antibody binding for detection. Secondary and tertiary structure are destroyed by this process. Additionally, SDS binds the proteins and thereby covers the chemical charges, leading to equally negative charged proteins. Therefore, the following separation happens solely by the size of the polypeptide change in the polyacrylamide gel. This is a chemistry technique that combines the physical separation capabilities of liquid chromatography with the mass analysis capabilities of mass spectrometry. LCMS is a powerful technique used for many applications which has a very high sensitivity and selectivity. Generally, its application is oriented towards the general detection and potential identification of chemicals in the presence of other chemicals. The statistical analysis and generation of graphs were performed by a series of software such as GraphPad Prism, and the differences were analyzed using ANOVA analysis, which is a collection of statistical models used to analyze the differences between group means and their associated procedures.
The Western blot with an ethanol diet induced an approximately 2-fold increase in the protein acetylation. The same Western blot analysis with propanol lysine antibody revealed mitochondrial protein propionylation also increases during the ethanol diet. No changes were observed in the mitochondrial protein butyrylation, and overall butyrylation signal was low. To test if alterations in the level of mitochondrial sirtuins could be mediating the changes in protein acylation, the total mitochondrial SIRT3, SIRT4, and SIRT5 were measured. There were no changes in SIRT3 protein expression in response to ethanol ingestion. Additionally, no changes in SIRT4 were observed. When measuring mitochondrial SIRT5, it was found chronic ethanol consumption did not change hepatic SIRT5. in contrast to previous reports of its reduction in rats. Together, this data suggests the changes in protein acylation were not due to changes in mitochondrial sirtuin expression. Type mice were fed an ethanol diet or control diet for 1 week, 3 weeks, or 6 weeks, and were measured for changes in hepatic mitochondrial protein acylation by Western blotting. There was an increase in mitochondrial protein acetylation with prolonged time on an ethanol diet. Furthermore, a time-dependent increase in mitochondrial protein propionylation was observed, which paralleled the increase in mitochondrial protein acetylation. When measuring mitochondrial protein succinylation, there was a time-dependent decrease in this modification. In addition to mitochondrial protein acetylation, nuclear protein acetylation has been shown to increase in response to ethanol diet feeding in mice. There's a possibility that other acyl modifications were altered in the nucleus in response to ethanol diet feeding. Similarly, changes in acyl modifications were observed in the nucleus as in the mitochondria. Finally, no large changes were observed in the nuclear protein succinylation between standard diet and ethanol-containing diet. Mitochondria and nuclear extracts from mice fed a standard diet or ethanol-containing diet were purified and subjected to the lysates to immunoprecipitation using antibodies. In the liquid chromatography mass spectrometry analysis, 22 acetylated proteins were found in the mitochondria and 19 acetylated proteins were found in the cytoplasmic and nuclear compartments. For the succinyl lysine antibody, 20 proteins were found in the mitochondria and 16 were found in the non-mitochondrial compartments. These results reveal an increase in the number of known modifications on hepatic mitochondrial and non-mitochondrial proteins, and it demonstrates that these new acyl modifications are abundant in mammalian systems. This diagram taken from the report is a pathway analysis of acylated proteins.
Previous studies of ethanol ingestion have shown protein acetylation to occur. In this study, acetylation is also shown to occur, but there were also new protein acyl modifications that were identified changing during ethanol diet feeding. In different cell compartments, the regulation of this occurs differently. Within the mitochondria, there were increases in protein acetylation and propionylation, so SIRT-free was tested for the observed increase, and there were no significant differences, suggesting that the increase of propionyl lysine during ethanol feeding was not due to reduced SIRT-free activity. However, during ethanol diet feeding, mitochondrial protein succinylation was reduced in contrast to the increase in protein acetylation and propionylation. This could be causing the modification to be removed due to an upregulation of SIRT5, although further studies must be carried out as there were no changes in SIRT5 protein levels in the mice. Similar changes in acylation of proteins occurred in the nucleus to those in the mitochondria during ethanol consumption. Protein acetylation and propionylation increased, succinylation was reduced, but butyrylation was unchanged. However, within the nucleus there is not a lot known about acyl modifications, and again more work is needed to understand the physiology. During ethanol diet feeding in wild-type and SIRT-free KO mice, there were no significant changes in acyl modifications within the cytoplasmic and microsomal compartments. However, the modifications are present and could be significant in a changed metabolic environment. To summarize, the study has been able to show post-translational acyl modifications in hepatic mitochondrial, nuclear, cytoplasmic, and microsomal cell compartments. During ethanol consumption, independent of serotonin expression, the acyl modifications are subject to change. More studies are needed to identify mechanisms leading to acylation and the specific sites of acylation. Finally, we can conclude that in addition to protein acetylation, protein acylation plays a key role in the metabolic regulatory network and is a rising mechanism throughout constant ethanol ingestion and metabolism.